Now, I would like just to focus with you of the three main classes of metabolite, terpenes, terpenoids, and also the others, in order to show you why they are interesting and what are their structure, chemical structure. So, firstly, one of the bigger classes is terpenes and terpenoids, and you know probably some of them like limonene, for example, or different geraniol or different other molecules that are currently used in different applications as food system or drugs or cosmetics and different things. So, strictly speaking, terpenes are hydrocarbons, okay, but they have a different possibility to be modified, or by alcohol, glucose means sugar, okay, or different, they may have some different modification to lead from, to produce some ether, aldehyde, ketone, carboxylic acid, and ester. I don't know if you are familiar with this type of classification of chemical molecules. Not so, not perhaps all. Okay, I don't want to make some biochemistry lecture. It's just to give you the idea of the chemical structure of this molecule is not always the same. You have a base, I will show you that, and from this base you can have very different organization depending of the post modification of this first molecule. And all these terpenes are present. Some are very important. For example, they are constitutive. Example, sterols that are in the membrane, okay, of the cell. So they are obliged to be produced. You can have also some like lignin, okay. It is a component of the cell wall of plant cells, so they are usually produced. So okay. But after you have some that are produced only in some plants. For example, you have some terpenes that are often considered as a scent constituent because you can smell it, in fact. For example, terpene camphor, you know, okay, this molecule, menthol, and also lemongrass, which is in citrus, for example. And what it is interesting is that all of these molecules are well known and well used in several applications, such as in perfumery, okay, between what we call essential oils. Essential oils, it's just a mix, a complex extract, okay, of what we can produce from plants, but it is not a soluble extract or volatile extract. It depends of the way you make your extraction. So essential oils are not exactly the same as, for example, a soluble extract of the plant. You may have not exactly the same composition. This is why sometimes it is important. And the other have some very important roles, such as phytohormones and also all the vitamins, okay, that are important for our food, come from these terpenes metabolisms. Here are a list of the main one. You could take time to look at from which plant they come from. For example, limonene come from mainly citrus, such as lemon, for example. And here you have an idea. It's quite even high. I can't read my own slide on this screen. Okay, that doesn't matter. It can be used because they have lots of effect. Okay, that can be detailed here. And well, here I just wanted to show you the basic scaffold of several of these terpenes. For example, limonene and linalool, they are different because one have a rings, benzenic rings, and the second one is considered as a linear molecule. This modification of structure may have lots of influence of their biological activity. So this is why it's important to have an idea of this. And after, for example, for pinene and eucalyptol that are very well known and very well used, you can see that not only they have rings like limonene, but also in the rings they have some specific other type of modification. For example, here there is what we name an epoxidation, which is very efficient to give here also a new biological activity of this molecule. So means that when you have a metabolic pathway, all the intermediates may have some activity by themselves too, not only the last one. So intermediates of this metabolic pathway can be considered as a product that can be interesting also to produce in a large quantity depending on their biological activity. Ça donne le vertige. Okay. See, here also it's quite the same but more completed, where you have here different type of terpenes, their effect, and also their medical use. I'm so sorry because you can't see nothing here, but with my slide as PDF document, you will see. Perhaps here it's more easier to see. Terpenes can also produce some molecules, you see, more complex. So you can imagine easily that it is not in one or two steps that from a precursor like limonene, we can arrive to this complex molecule. This means that we have a very big metabolic pathway. And from here, you have some example well known, Taxol. So we have seen, okay, the history of the Taxol, but also artemisinin, with it is an anti-paludéen molecule. It's, I think, the only one that are very efficient. Okay, so it's important. And it is also an example in which we will have time to discuss, because these plants are able to produce. We can extract artemisinin from plants, but it can be also produced by biotechnological ways. And also in cannabis sativa, you can have some specific molecule considered as not so good molecule, but they have some very interesting medical property that can be also useful for several treatments. And gymnosid too are some molecules that are very interesting for their activity as antioxidant, for example, and then can be used combined with chemotherapy, for example, in cancer treatment. Okay. And the ginkolid also have lots of property, but you can see here the complexity of this molecule. Well, so what is the organization of the metabolic pathway? Not the metabolic pathway, but the class of these terpenes. You have a first molecule, isoprene. Isoprene is the base of all the terpenes, because after, it's just a question of polymerization of this molecule. So when you have just one isoprene, it is a class of terpenes named hemiterpenes. When you combine two isoprene, you have three different ways to make the assembly of the two isoprene. Means that you can here introduce a first diversity, okay, molecular structural diversity that will lead to several other types of metabolites. So condensation of two isoprene lead to what we call monoterpenes. Lots of monoterpenes, such as limonene, for example, I show you, are useful for several applications in our lives. And after, you can add one and one and one more, so you can define what we call sesquiterpene that have three isoprene motifs, so means they have 15 carbon, okay, but because isoprene is 5 carbon. And diterpenes, cis-terpenes, triterpenes, tetraterpenes, that are the more known and the more useful terpenes, okay, in the world. And after, we can go in what it is more and more. For example, the latex, okay, the rubber. In rubber, you make some latex, and this molecule have a very high number of isoprene condensation, more than 100 and 200. So from just one small molecule, okay, we can introduce a very large amount of diversity of terpenes. And all of them have some biological activity. So here, for example, it is one of the more known, okay, metabolic pathway that lead from two monoterpene, okay, that have been a little bit modified because they have two phosphate. It is isopentenyl diphosphate and its isomer dimethylyl pyrophosphate. And when you add both of them, you create a first monoterpenes, what it is known as geranyl pyrophosphate or diphosphate. And when you add a third one, you make sesquiterpenes, such as farnesyl biphosphate. Okay, it's an example. And you can add another one to produce some geranyl diphosphate. Why I show you this example? It is because it's these three terpenes are involved in a several number of other pathways that lead to lots of terpenes in the cells. And in particular, some of them can be combined 2 by 2. Example here, 2 farnesyl diphosphate lead to squalene. And squalene, I will show you, it's a very important molecule that it is used in cosmetic. And why it is named squalene? Because it was found in squal, in the animals, in requin. And in fact, they have fishers have taken lots of this animal just to isolate squalene. So you have understand that if it is exactly the same that when it is a tree that is able to produce some, when it is an animal, you can put them in danger if you collect them with a non-controlled system. So now it is one of the products that is processed via metabolic biotechnological system, particularly in yeast, for example. So now it's all the squalene use come from biotechnological resource. And it is exactly the same for the phytoene, the cis-phytoene, okay, which is a combination of 2 geranyl pyrophosphate. Don't be afraid. Okay, it's just to illustrate what I want to show you is that in fact here you have what we call linear, okay, linear terpenoids, even if they are designed like something quite with a cycle, but they are linear, okay. And from this linear, after you can shift to cycle 1, okay. It is what I have shown you, and after from this cyclized molecule, you can combine them, okay, in order to have molecule more complex, leading to diterpene, cis-terpene, triterpene, and so on. Some stay, like here, linear. I show you, for example, a squalene and so on. But some become with not only one cycle, but multi-cycle. And cycles are very interesting because they are able to induce specific biological activities, in particular cytotoxicity of the molecules. So this is very important. Here, it's for you. You will see a time, because here I have made some specific example of each type of terpenes in order to lead to these polyterpenes, more than 40 carbon. I explain you the natural rubber, and also perhaps you know this plant Gutta percha. No, this Gutta percha. No, sorry, is able to produce a specific exudate from leaf that was very elastic. So it was used initially for dentistry, to develop several things for dentistry. Okay. It's a pity because I don't see nothing. So here, just to illustrate what I said to you before, from the linear one, you have the cyclization. And after, when you have cyclizated the molecule, you may have some functionalization by addition of different groups, alcohol, and you can make some double link between 2 carbon to conduct, and with oxygen to conduct to aldehyde, the ketone, and so on. And this functionalized molecule are always directed to a specific activity in the cell. And for us, it's very interesting to have an idea of this. And also another point, it is the stereochemy. Okay. In the nature, when you have a molecule and when you add, so for example, a carbon or a different group, if you have, for example, a ring, the group can be added on upper the ring or down under the ring. And this is what we call an isomer. And two isomers may have completely different biological activity. So in the nature, the enzymes that are able to produce one isomer is not the same as the enzymes that are able to produce the second one. But in the nature, very often you find the two isomers in the same cell. But for us, when we are interested by just one of them, we don't want to produce both of the two isomers by a biotechnological process. So we need to find which enzyme is able to produce one isomer instead the other one, in order to address directly our production to the metabolite we want to do. And unfortunately, lots of terpenes have isomer. So we need to take this also in account when we want to develop a process of production. Okay? This is also the same for other monoterpenes. I speak here about iridoids. Iridoids have this type of structure, very simple one, but you can add so many different things that lead to complex, very complex terpenes. And for these terpenes are often combined with other type of secondary metabolites in order to produce alkaloids. One of the heart of some alkaloids are iridoids compounds. So they are very important. And also they are cyclized and functionalized too by several other modifications, including carboxymethylation, glycosylation, acetylation. Sesquiterpenes, as I said to you, are very important for essential oil. Here are some examples. And also they may have one or two cycles depending, and also they can be functionalized by several different modifications, in particular methylation or hydroxylation. And for example, here you know the tree that gives some santal wood. Santal, yes. And the compound that gives this particular odor is known as santalol. And santalol is a sesquiterpene, that it is why you can smell it, okay? And it is a very important molecule that now can be produced by chemical, organic chemical process, but also by a process, engineered process. This is another type, okay, I go fast. And also, one of the type that it is very important, it is what we call lactone sesquiterpene. Lactone, you have here, it's a process particular that gives this particular structure of two oxygen, and they have very important antibacterial and antiparasitic activities. Artemisinin come from this type of structure, and in fact, lots of these molecules are tested as antibacterial one. So in the family of terpenes, these one are very interesting to study and also to characterize according to their biological activity. And also in this case, you have a different structural modification of variation, okay? And also you can have addition of several other polycarbon molecule or several alcohol motifs. But don't forget this type of molecule for development of process. Diterpenes. Diterpenes, they start to have bigger molecule, okay? Diterpenes mean 20 carbons. And the most known is chlorophyll. You know this, every one of you. It's one of the more important one. And also some phytohormones such as gibberellin are part of this group of diterpene. They are not really used for several applications, but other type of diterpene have a very important activity, such as taxol. Is come from this type of subgroup, and you have followed the story of the taxol with Professor Smita. And it is not the only one. There is other type of diterpene that are very important, and unfortunately, they are produced only in a specific part of the plants. For example, taxus, it was only in bark, not in stem, not in leaf. It will be interesting in leaf, but not in the bark. So by this way, the sourcing of this molecule will be always a problem. So we need to find a solution. One of the solutions was to try to find some productive hybrids. So people have tried to cross over several specimens in order to produce some that are able to produce more molecules. But there is a lot of limitation. A second way, which is used for lots of anti-cancerous molecules, is to take some intermediates in the plants that are produced in a very bigger amount than the final product, and to make some semi-synthetic molecules from these two precursors to produce a final one. By this way, we increase, the amount of the final molecule with a very, very important way. So one of the solutions is to find intermediate plants that are able to produce a lot of these intermediates. That was also what we call the taxane, for example, are produced by this way. Another way is a biotechnological way, cell culture, endophytic fungal culture, for example. And now, what I would like to explain you at the end of this lecture is the way to produce them in yeast. So there is now several new way, to produce this complex molecule or their precursor, in yeast in order to have enough material. Triterpenes, what it is interesting, it is this group, the steroids, that are very important for several control of physiological activity of the cell. So they need to be produced in a big amount. So it is also a question, so how to have them. What it is interesting is that, in fact, some are linear, some result from cyclization. Squalene, for example, can be also cyclized in epoxy squalene that can have lots of property, very important property in cosmetic formulation, for example. Steroids, I say to you, are very important, like cholesterol. And you see, depending of the degree of cyclization, 3 or 5, 5 years, pentacyclic is 5. You may have some complexity of this molecule. In particular, these molecules are important in the cuticle, of the plants. This is a particular thin layer of that cover all the plants to protect the plant from transpiration, so means loss of water, and also it is always a way to protect them from UV. So in this particular cuticle, we can find lots of molecules that have ability to protect against UV. And in cosmetic, to produce some specific cream, including this type of molecule, is of first importance due to the change of climate that, increases the demand of this type of product. So this is why, it's interesting to see the composition of this cuticle and also the molecules that are in this type of cuticle. Tetraterpene, I don't know. In this case in particular, focus on the beta-carotene. Beta-carotene is considered as a vitamin. It's important. This is why we say to child, please eat a lot of legumes, but because they include a lot of carotene, and it's a way to protect themselves ourselves for different disease. And in fact, this beta carotene is a start point of several other family of terpenes that are of first importance. So this is interesting also to have this knowledge, okay, in this type of molecules. And in this group, there is also the phytohormones abscisic acid, that it is very useful for plant cell elicitation or entertainment, okay, because it's a phytohormone that lead to lots of signaling, cellular signaling, that can also lead to produce other type of secondary metabolite. This is a link with all the pathway I explained before. And finally, the polyterpene. So always today, natural rubber is collected from the Hevea brasiliensis, okay, the tropical trees. You can see now it's completely regulated. There is some plantation dedicated to this, so there is not the risk of the rarification of the plant. And gutta-percha was also a latex that comes from a particular Paliquium gutta. It's a plant that now is completely protected, okay, because they have been used a lot. And in fact, now this question is less important because synthetic polymers are produced in a low cost. So it is not necessary to produce another type of alternative way to produce them. This is important to have in mind. It is not necessary to engage programs on molecules if the cost when they are produced by a chemical way is not important. Because for industrial, okay, opportunity, it's not necessary to engage, okay, biotechnological process if chemical ones are sufficient, which is the case here. I think you don't know what it is, a gramophone record. It's a long time ago, and the disc were produced with this type of material, okay, instead of vinyl, which was synthetic one, and now it don't. See, there is a new life for vinyl, actually. Okay, this is just to show you can. Don't worry. I have left here the link. You can upload this. It's free. It is a very interesting map. This is a zoom, for example, where for each type of terpenes, from monoterpenes to polyterpenes, you have the list of the plants that are able to produce specific one. And here you have also the particularity of their structure, which is very important because it is done from monoterpenes to polyterpenes, and for each category you have this type of description. So it's very interesting that someone have done this works for us, because by this way we can also have an idea of which are some terpenes that could be interesting also to manage in biotechnological approaches for production. So don't hesitate to take a look of them. And depending of their scaffold, that is easy to obtain or difficult to obtain, means that it will be not exactly the same program we need to engage to produce them. Okay. And to finalize with terpenes, what are the applications? There are lots of a large set of applications in perfumery and cosmetic as monoterpene, I explained you before, and also two molecules that are very important, geraniol, particularly, and limonene. They are in quite all cosmetic products. You can see that in the description of the product you use. And lots of plants produce geraniol, but it is always not sufficient for the demands. It is a real increasing in demands. And saponins. Saponins are very important. They are used in cosmetic, but they are also used in pharmaceutical formulation, because as it was explained, it is some molecules that are able to modify the permeability of the membrane, the cell membrane. So, for example, it can be used as adjuvants in pharmaceuticals in order to let them more efficient to enter in the cell. So there is a lot of programs dedicated to this specific type of terpenes family. In food technology, carotenoid as vitamin is obviously one of the most and bigger targets. Okay, in pharmaceutical industry, the list is very long. Okay, of the drugs that include this type of molecule, and lots of have been approved as FDI by the FDA, means that they can be commercially used by pharmaceutical industries. So it's very important, and the demands are always increasing, and also in the dentistry. And insecticide also is a big part of the market using this type of terpenes, because in fact they can be used as a repulsive. You know that what? Sorry, repulsive. It's a French word, but I have not the English word. Means when an insect is coming, instead of stay on the plant, it is okay. Repending. Okay. They are used as repending. Thank you. Insect repellents. Yeah. That's good. Okay. Concerning phenolic compounds. Phenolic compounds, you know them also very well because all of them are in your food. Okay? You in the spice, okay, you use for your very good food and good cooking, but also in wine, but also in the tea. There is a lot of phenolic compound in the tea, and you know that because in fact they have some very benefit for our health, means that they are of first importance. It is also one of the largest with the terpenes classes of this metabolite, secondary or specialized metabolites, and about 8,000 molecules have already been completely characterized and reported as phenolic compounds. And what it is true is that each year new compounds have been characterized and increase this list. Means that in fact there is a lot of works done over the world on this specific family of specialized metabolites. So the phenyl compounds derivates from the amino acid phenylalanine. We will see that after the metabolic pathway. And in fact, it's interesting because it is also this type of molecule we find in each organism, and particularly in algae, means that in fact they appear in firstly in aquatic plants, which is not the same case for example, all the terpenes, some yes, but not all, and also we will see that for nitrogen compound such as alkaloids. And they are really close linked with the terrestrialization of plants. So it is important to compare, for example, mosses with angiosperm to find several different types of phenolic compounds. They contain about 40% of the organic carbon. And what is the main compound that that accumulate a large part of carbon in plants? Do you have an idea of? Yes, what is, according to you, in the plants, what is the molecule that is a phenolic compounds that is for a large part responsible of this accumulation of organic compound? It was one of the bigger resource of organic compound. Benzil? You say? No.
But the question is that which is that molecule from this class of compounds most abundantly found?
That is responsible for a large part of this.
40% of
okay. It is what is used, for example, as fossil carbon, for fossil carbon. From what type of molecule derived this? It is a molecule that is responsible to make the wood. Lignins. Okay. It is one of the most important molecules of these phenolic compounds because by itself, more than 30% of the accumulation of carbon. So this is important. And lignins are very interesting also to study and also to produce for several other applications. And as for terpenes, the first molecule was isoprene. For phenolic compounds, this is why we call them phenolic, it's phenol. Okay, phenol is a very small molecule with a ring, okay, and benzenic rings, and also with an hydroxyl ornamentation. And from this phenol, you can synthesize all the polyphenol, okay, that you can identify. Here are the different type of polyphenol, such as flavonoid, tannins, phenolic lignins, and also stilbenes that are mainly present in grape, okay, that are very important. You know, perhaps resveratrol. Resveratrol is one of the main stilbene known, and it derivates from this type of molecule. So what characterizes all the phenolic compounds is the presence of this, at least of one, means that you may have more, okay, phenolic nucleus. And how he organized this world of phenolic compounds into main branch, what it is called simple phenols, in which you will find phenolic acid and the coumarin, and specifically some of them can be also transformed in furocoumarine. Furocoumarine is not put here, but they are very important for several pharmaceutical applications. And for phenolic acid, you can see here lots of molecules, namely hydroxycinnaminic acid, that are very important. You can find some in coffee, for example, such as caffeic acid, which is known also to be interesting for complementary food production, for example. And also hydroxybenzoic acid. Vanillic acid, for example, is part of this. And it is a small branch compared to this one, what is called polyphenol. Okay, polyphenol means that you have not only one phenol molecule, but more than one. And in this case, you have primarily division between flavonoids and non-flavonoids, quite easy to see. And in flavonoid, you have this different type of subclass of phenolic compounds: flavones, flavonols, isoflavonols, flavanols, anthocyanins, and chalcones. Okay. We will have time to have some example during my talk. I don't want to decipher all of this, just to show you that it's very complex organization of this particular family. It is what we call flavonoids. And in the non-flavonoids, you have tannins. Tannins, it is what you find, for example, in wine. I don't know if you have tasted some wine sometimes, but it became from grape. And it is what it gives you a sort of acidic sensation of the tooth, okay? And in tannins, you have hydrolysable, condensed, and complex, okay, molecule. There is not one, it's a big family. And in non-flavonoid, you find lignans, okay, in which you have also lignans, and also the stilbenes, what I explained before, including the famous resveratrol. Resveratrol is one of the molecules very interesting in several cosmetic applications, but also as drugs, because they have some anti-cancer activity, antioxidant activity, I don't know how many biological activity have been characterized with this molecule, for example. So here it is exactly the same. I don't want to detail that, just for you to see how we classified this type of molecule. You have the simple phenol, that's how just one, phenol with different modification, 2-hydroxylation, 3-hydroxylation, for example, but you may have some methylation and different other modification in this big family. And after, you have some combination of these phenol molecules with different other carbon, or 1, 2, 3, or 6 means that you have 2 phenols, for example. And after that, you have several types of combination. And this is the basic skeleton that lets to us to make the classification of the molecule. You have to realize that the more they are complex, the more the metabolic pathway leading to the molecule is evidently complex and long. Okay. Here are some examples. Okay, sorry. I have to find a way to change some of my slides for the next time. So here are the simple presentation of the origin of the phenolic compound. We will see that they come from Chikimat pathway, one of the 4 routes I showed you before. And this leads to form Chikimat that gives the first phenolic acid, the simple one. But in another side, this Chikimat pathway can be connected to the phenylalanine pathway that lead to the production of phenylpropanoids. And when you have the combination of both, you can increase the variety of scaffold, of this phenolic compound. And what it is also interesting is that mevalonate pathway lead also to specific complex phenolic compounds, such as flavonoids, stilbenes, and this one. You see that the structure, And what it is also interesting is that, in fact, from this one, you can have also polymerization process that give not only simple molecule, but real big molecule, in particular that leads to lignins, very complex molecule, and also lignans and different others, such as a part of tannins. And you can have combination with some of oligo or polymer with other flavonoids, for example, to have more complex molecule. It's not my purpose is not to show, for you to learn of this. It's just to show how it's complex when we want to identify just one molecules, because when the plant is able to produce a lignin, means that this plant is able to produce lots of intermediates. So when you make some extraction, it is always a complex extract with more than 100 or 200 or 300 different molecules inside the extract. So when you start some screening for a campaign, it's really difficult, because to know which molecule is able to have a good biological activity, theoretically, you have to purify each molecule, which is completely impossible to imagine. So it means that we need to develop several strategies by what we call fractionation, make some fraction of the total extract in order to see if some of the fraction have more activity than in other. And after, in a fraction in which we find a good activity, we start to make sub-fractionation, and we do the same. And in a sub-fractionation, we can, after, depend on the number of the molecule, make some purification in order to arrive to the molecule that could be interesting to consider as the only one. So, and this is because in the pathway, you have lots of intermediates, Example of flavonoids that are well known. I show you different concerning tea and wine, but also you have some spices, curcumin that come from curcuma, and so genistein. It's a flavonoid very important in soybeans. This is why they are very useful for food, complementary food, and so on and so on. There is so many applications. Okay. So far, about 10,000 flavonoids, non-flavonoids, polyphenol, okay, if you have the scale in mind. So it's just one part of this word, okay, of phenolic compounds. So only 10,000 have been already identified. More than, quite 50% are monomer, dimer, or polymer. The other are a combination with something else. Precursor is chalcone, that it is catalyzed by a specific enzyme, chalcone synthase. We will go to the enzyme after, okay? And in particular, some of them are able to be linked with the carbohydrate, mainly glucose. So glycosylation, which is a modification of phenolic compound, is very important. Why? Not only because it gave different other biological activity, but it is also a way in the cell to accumulate this molecule in a particular organelles. Okay, for example, vacuoles, for example, nucleus also. They can accumulate glycosylated specific metabolite in order to protect them from other enzymes that will be able to cut, to destroy this molecule. So glycosylation is very important. And this is why, largely, when we make some extract and when we analyze the extract, we find metabolite specialized glycosylated. Okay? But sometimes the best activity is the non-glycosylated form of the metabolites. So it means that we have to make, okay, a sort of balance between both. Here are some examples of structures that are known for different type of classes. It's not important, just to see that there is not so many. There is a lot of work to do if we consider that more than 10,000, but I think more molecules are from this class. Okay, so example is here of flavonoids, in which you can find the different subclass of flavonoid. Here it's very interesting table I wanted to share with you, because there is a color code in order to know why we name them flavon, flavonol, flavanon, etc. Because you see, flavon, it is because there is this particular modification, it's in red, and from this flavon you can add an hydroxyl group that comes from flavonol. Okay, so by this way you know what is the modification. And also here you have some other rearrangement of the two rings, okay, that come to flavanon, okay. And also here you have addition of modification here of the ring. And if we can go by this way, flavanol 3 is another type of organization, and this is exactly the same for this. So it is just for you, when we will have time, to see which is the scaffold of the molecule according the subclass of each, okay, flavonoids. Because it's a very big family that are very interesting to explore for bioproduction. And here, we will see that after, just for you to have in mind, this is, for example, the flavonoid biosynthetic pathway. You can see that you have a common part of the pathway named phenylpropanoid pathway that lead to phenolic compounds and non-phenolic flavonoid and non-flavonoid compounds. But the intrinsic is here, okay, catalyzed by one enzyme, chalcone synthase, okay, that is able to produce a chalcone. It is what I said to you. You see that this enzyme is able to condensate one molecule of coumaryl-CoA with 3 molecules of malonyl-CoA. Okay, this is the first step to entrance in the metabolic pathway of flavonoid. And after, from this precursor, you may have several modifications. Here they are illustrated. I don't want to describe the whole, but what it is interesting in this slide is that each modification is catalyzed by a specific enzyme. Here are the initial of the enzyme that have been characterized to make this modification. So it means that, for example, I wanted to produce this molecule in a cell or in a yeast cell. I need to know all of these enzymes. I need to have all the sequence, all the gene encoding this enzyme if I want to rebuild this metabolic pathway in another cells. I mean, I need to introduce the genes in the genomes of my cell host, okay, in order to produce the enzyme to modify and to rebuild the metabolic pathway in the yeast cell. So this is why it is important to know exactly what are the different chemical structure, because with chemists, we talk with them and they explain us, okay, if you have this molecule and you want this one, you have an hydroxylation, you have an epoxidation, you have different type of modification, and with them, we imagine what are the routes that can be used chemically, biochemically, okay, from a precursor to a final molecule. And by this way, it is for us an indication of what are the enzymes we need to know that are able to do exactly this chemical modification or biochemical modification. And sometimes we may have 3 or 4, 1, never. At least 2, 3, or 4 different pathways possible from one molecule to a final one. And this is the way for us to imagine what are the enzymes that could be involved. And here, some of them have been, lots of them, okay, have been already identified, but depending the final molecules, lots of are needed to be identified today in order to go specifically to this phenolic compound instead of this one, for example. Okay, don't be surprised. It is also here what it is useful, the most used of this flavonoid in pharmacological application. Here are the main activity: anti-inflammatory, anti-cardiovascular, antibacterial, antioxidant, and so on. Here are the classification between this flavonoid and the name of the flavonoid, and here the source of plant in which it can be used, found, sorry. Okay, so by this way you could have an idea of where you can find this type of metabolite, and this illustrated what it was said by Professors Mita and I, is that one plant is not able to produce all the phenolic compounds, okay, just a part of the possibility. Okay, another example is anthocyanins. It's interesting because here it's the basic skeleton of anthocyanin, on which you can add several group, carbo or not carbo group, hydroxyl or methyl or methylated, and this can give lots of different type of molecule, and also you can put some carbohydrates. I said to you, glucose, rhamnose, xylose, galactose. This is some glycosylated, okay, molecules that are very interesting also for several other applications. And here also the plant origin of the molecule. It is also important when you have some specific anthocyanin, not all the plants produce the same anthocyanin. They are red at the final, all of them, but the color of red depends not of the same anthocyanin each time. Okay, because anthocyanin for the plant is not only to make them red. Okay, it's mainly because it's some phytoalexin. They are considered as some phytoalexin because they protect the plant several, mainly from predators, herbivores, for example, mainly. it's quite interesting. They attract them, but after they kill them. So it's not a zoological system in plants. They are very clever, okay, to survive in this world. Lignan and neolignans, they are not lignins, but what it is interesting is that they have been a quite simple molecule, phenyl, you can see here, plus 2 other carbon. And from this phenylpropanoid unit, you can have some polymerization, here dimerization, depending of the sense of dimerization, and you can produce lots of molecules named lignan or neolignan. And lots of lignan have anti-cancerous activity. It is the example of etoposide that it is produced by podophyllum, for example. And at this time, these molecules are not produced by chemical chemical system. So this is why it is urgent to find a way to produce them, for example, before the plant will be disappear completely in Himalaya. Okay, even if Himalaya is a big geographical zone, it can be a problem for the sourcing at the moment. So this is one of the molecules that is very important. And here some other, modification of the first. You see here the first unit. It's just for you to illustrate, okay, the variability of the molecule we can find. So means that, for example, if I am interested by this one, I need to imagine from my first this one from this first scaffold, what can happen to lead this to this particular molecular structure. Because if I want to develop some biotechnological process to produce this one, I need to identify the enzyme, not only one, probably more than 2, 3, or 4, that could be able to gradually modify my precursor to produce the final molecule. Okay. Okay, this is the application of several lignans and neolignans, anti-cancerous, as said to you, etoposide, but also for different other diseases, anti-inflammatory, anti-oestrogenous, anti-HIV. Lots of them have found very interesting activity against the virus, etc., etc. So please take time to look at them. And to imagine, okay, the complexity of their structural composition. What I have not said is that sometimes it's not only one molecule that is efficient. In the cell, it is the synergy of 2 or 3 specialized metabolites that may have the best biological activity. So sometimes it is also interesting to see the process of the biosynthesis pathway, because not only the last products, but with some intermediates can have a bigger biological activity than only the last one. So this is what also it is interesting to have in mind. Okay, this is another one. This is just to see that with this, in particular proanthocyanidin, they are produced by a very high level of isomerization that leads to very complex molecule in case, for example, of tannins. Okay, I stop with my example, just the application in perfumery, in cosmetic, in industries, in particular phenols. The simple phenols is very important for plastic industries because it enters in the composition of several plastic, including bisphenol A. You know, it is used for several plastic objects we use every day, and also in phenolic resin. And different other manufacturers used also different other phenolic compounds, such as this one, and that can be used for several applications not in pharmaceutical, ni cosmetical, because in pharmaceutical, phenol was also by itself used as an antiseptic, but in fact now it is replaced by the other ones that are less with a secondary effect, because phenol has lots of secondary effect on our health. But there is also a different, I have not made the list here, but I have spoke about some, okay, before, as anti-cancer and so on and so on. Etoposide is always used, okay? And as a Taxol, the demand is increasing and the production is not quite stabilized, but sometimes it's decreasing. So there is something wrong between the two demands. Okay, so we need to find alternative way.